How is it that we find ourselves surrounded by such complexity, such elegance?
The genes of you and me, the genes of you and me, are all made of DNA. We're all made of the same chemical DNA. We're all made of DNA.
Hi, you're watching DNA Today, a multi-award-winning genetics podcast where we explore everything to do with genetics, from CRISPR to rare diseases to new research. We have won the Science and Medicine Podcast Award for many years now. We have hundreds of episodes, and we really hope you enjoy these conversations where we dive into so many genetic concepts. I'm Kira Deneen, a certified genetic counselor, and your host. We are delving into the genetic causes of epilepsy in this episode. To do so, we're joined by three guests. Dr. Kari Mattison and I connected as she is a listener of DNA Today and shared her research with me. And we got to talking and we're like, oh my gosh, we got to make an episode out of this. So that's what's happening today. She focuses on the role of ATP6-VOC in epilepsy. She earned her PhD in genetics and molecular biology from Emory University, where her dissertation work was on functional evaluation of novel variants identified in early onset epilepsy. And again, we're going to be talking about a specific paper, which we're going to link to the show notes there. Dr. Luis Miguel Oliveira and Ana Rita became patient advocates after their child Daniel was identified to have a pathogenic variant in the gene that Dr. Mattison studies. Dr. Oliveira co-founded v-ATPase Alliance, a nonprofit dedicated to finding a cure for rare disorders caused by v-ATPase genetic mutations, where he brings his expertise from working at the Michael J. Fox Foundation's research programs teams. So thank you so much, all three of you, for coming on and educating me and the audience, not just about early onset epilepsy, but this very particular type. And as listeners know, we cover a lot of different rare disorders on the show. So I'm really grateful that you guys felt comfortable to come on and educate everybody. So Dr. Mattison, I thought I'd start with you of giving us an overview of your research, and you'll have to, make sure that I'm saying it right. ATP6V0C or OC. So break that down for me a little bit. Correct me.
Oh, written, it's a 0. We tend to say O, kind of like we would with a phone number where you say like 208 or something. But a lot of times, and what you'll hear me say a lot, is we just call it VOC because the full gene name is a mouthful otherwise.
It is, as you guys can tell.
And that's enough
as I'm saying it.
Yeah. Yeah. So if you hear me say VOC, I'm talking about the gene. Same way with the complex it's part of, we call it the v-ATPase. The full name's the vacuolar ATPase. Again, long, unnecessary kind of names, but they do serve a purpose in terms of broader genetics.
Very cool. And what drew you to studying this v-ATPase complex and its role in neuro disorders, especially like in the pediatric space?
Yeah, so my dissertation lab was an epilepsy lab, and my PI was mainly focused on sodium channels, but we kind of had this offshoot of looking at genetic testing data we had gotten from Emory's in-house lab. So it was de-identified to us. Literally everything we got was just a sequence information, a 5-digit number. We had to go through like 3 steps to get any patient information. But with that collaboration, right, we had this wealth of information where we could go through and say, okay, this is a variant that looks like it could be pathogenic, for XYZ reasons. So is it expressed in the brain? Is the, nucleotide change going to amino acid change seem pathogenic? Doing a lot of this kind of, I called it like, it's like treasure hunting, but you're also like Sherlock Holmes, all in one, trying to figure out this information. And so we had that wealth of information. I knew I was more interested in the human side of epilepsy because our lab had a mouse component to it. So that's where I started with this genetic testing data. And it really kind of boringly spurred out of the need for me to need a project to defend for my second year oral qualifying exam. And so we were coming through this data and we found this patient with a variant in VOC. It's really meeting all this criteria of, well, this looks really interesting. And then I dug some more. And back in the, 80s and 90s in yeast, believe it or not, people were studying the vacuolar ATPase and VOC specifically. And so that really piqued my interest of, okay, there's something we can do here functionally where if we can find more patients than just the one we've identified, we could really do something here that's helpful and, say, how much function do they have with these variants? How does it impact the v-ATPase? And that helps provide, another level of, okay, this variant really does seem like it's pathogenic, and gives insight too, right, to kind of the treatment that might be needed or useful, if that makes sense.
And it seems like there's a lot of variants that can be pathogenic and basically make something not work the way it's supposed to, or in a way that makes the pathway correct and functional. So with all of these different variants, do they affect the function differently? Like, what is observed with that genotype phenotype correlation? Or is it, if there's any mess up, it's going to lead to epilepsy or a much higher risk of epilepsy? How much do we even know to answer some of these questions?
So I guess to date, now I have to remember how many patients we had in the paper. I believe it was 27 we had total. Woo! I'm getting thumbs up from everyone else. And we did not test all of those patients. So just a quick insight into how publications work. We had the manuscript written and we're getting people emailing us say, hey, we have a patient. Well, we no longer had time to do the functional tests and create what we needed to do it. So if you're wondering why there's some variants that aren't functionally tested in papers, that's usually what happens. Is we thought the benefit of getting the paper out with what we had was better than waiting to get extra information in there in terms of the testing, because we had a pretty, I guess, broad span of what we tested. In terms of, your question of genotype phenotype correlation and how they impact the v-ATPase, we saw that all the variants acted in a manner that we would call loss of function. So they essentially decrease the function of the vacuolar ATPase, right? So they're saying at some level it may work, but it's not going to work as efficiently. And so that can cause downstream effects with just, cellular energy needed to try and have it do its normal job, but also in some cases it can't do its job at all. It's kind of the best way to explain it. We never say never in genetics. So there is a chance potentially we could find a variant that's called gain of function. So all of a sudden the vacuolar ATPase, instead of doing its job at I guess the analogy that comes to my head is like one full-time equivalent or one full-time employee, all of a sudden it's working like twice as hard. So it's like one person doing the job of two people, right? I do think that is potentially less rare because we're talking about a complex comprised of 13 subunits. And so this is one gene that encodes for one subunit, and the other subunits aren't going to necessarily be affected. So gain of function, possible to me with what I know now, seems less likely. Yeah, so these variants are basically taking the complex, making it so it works less efficiently. I guess a couple other things that would be important to note is that there are 9 copies of VOC in what we call the C ring. So it's not just one copy present, there's actually 9. And with that, patients are all heterozygotes that we found so far. So they have one variant or one mutant copy of the gene and one wild type or quote unquote normal, right? And so there's going to be some random assortment of how many normal versus mutant copies get into this complex. So that's something to keep in mind, is it's not as straightforward as it is if there was just one copy of this gene present in the complex. I guess one, yeah. And then in terms of genotype phenotype correlation, with what we had available to us, we didn't see anything. So we didn't see that patients were the variant we tested that showed close to 0% of wild type function versus those that maybe had like 20, 30. They didn't show really any difference in the clinical presentation in terms of what we had for information. So we mostly had, did they have epilepsy, and then seizure types? Did they have intellectual disability, and to what severity that was? And then we had information on developmental delay. So when did that hit, and kind of what systems was it hitting? But we didn't have anything beyond that. And I think Luis and Ana can go into this further with the Alliance, but one thing that we're really pushing for is deeper clinical phenotyping, because you might find something when you're looking at more phenotypes, but also when you're listening to the people that are living this every day, instead of kind of what I call a diluted clinical report from clinicians. I know in my personal experience, I read notes from my revisits and I'm like, this doesn't actually match what I said, but it's close, right? Um, so, there could always be something. It could be something that comes on later in life that we find might be where the difference is. But for now, in terms of the neurological dysfunction or disorders we're seeing, we don't really see a difference in terms of the functional level from the genotype corresponding to a difference in the phenotype, if that makes sense.
Yeah, there's just so much to learn within this. And really initially looking at data, you're like, okay, well, what do we have and what is the sample size? As you said, it was kind of growing as you were ready to publish, and you're like, okay, well, we want to include this, but we won't be able to have as much information. So there's a lot of real life implications that just happen in that point. And the way that you three connected is, Dr. Mattison, your paper, I was going to say was released, but it was published. And so you all connected from that. Tell me a little bit more, Dr. Oliveira, about that connection and just how your collaboration blossomed from there. And you might also have advice for people that are seeking papers that are behind a paywall too, I'm thinking.
Well, kind of. I think this was open access. But well, first of all, let me just thank you so much for having us. It's an honor to be on the show with you guys, and it's really a great pleasure to be able to talk about our journey, about our son Daniel, and this new organization we started. And it's now like intimately related to our sense of being in mission of finding treatments for him and the other kids with the same diagnosis. Dr. Mattison's paper came referenced in our genetic report, and it was really like our first glimpse of light in a very dark hour, I must say, because obviously the paper, with the paper came the diagnosis, and that was a huge punch in the stomach, because it's the realization that our first baby was not gonna have a fair start in life, and this was a de novo mutation, which is the case for the vast majority of children with this disorder, and this means that it was not inherited from any of the parents. It occurred by virtue of biology, if you will, and but on the other hand, Dr. Mattison's paper gave us a target and told us what was wrong, and that is really the first step to get it fixed, right? And I remember to send her a message on Twitter. Like, I found her Twitter profile, and introducing myself, shared our story, and she was very kind in her response, and helped us to connect with other authors in the paper, and sort of understanding who was doing what, who was doing more basic science work, who was doing more clinical work. And, without knowing at the time, these were really the first baby steps for the Alliance.
Yeah, that's amazing. And Anna, to loop you in, I mean, tell us a little bit about Daniel in terms of what led to, like, what symptoms were coming up for Daniel that led down to the steps of the diagnostic odyssey, and what some of those steps were before you got this report.
Yeah, so just saying the same as Louise, thank you so much for having us. It's a pleasure to be here and be in the presence of you all. And so baby Danny, so we call him Danny, not Daniel, but just Daniel is fine. But it's just the same kiddo, and he was a baby that when he was born he didn't cry, and he was a premature baby. And when when kids are premature, they do the glucose test where they kind of just keep on testing every couple hours. He never cried, and everybody was like, "Oh, you're so lucky," and we were like, "Are we lucky? Like, he doesn't seem to feel pain. It's but you can sleep because he doesn't cry." Yeah, but I don't know. It's weird. And this was Danny from the start. So he was the perfect baby. He didn't cry. He was always quiet. He didn't require any attention. He didn't. He was never fussy if he was hungry or dirty or nothing. He was just like perfect. And spoiler alert: there's no perfect baby. If a baby's perfect, something is wrong. And because dad is a scientist, we start talking with the pediatricians, and everybody was like, hey guys, stop reading. Your baby's perfect because, he's in the good percentile. He's growing. In terms of growing, everything is fine. Um, he's not very good at the breast, but you can fed him a bottle. He takes a bottle. So, he's just lazy. So we kept like hearing this and keep pushing it to like something is wrong. We know something is wrong. We can't, explain what it is, but something it's not right. And this kept going for a few months, and then between the 6 and 8 months, Danny had an oral regression where he was already saying a lot of consonants when he was speaking, and at some point in a matter of days he lost all of the sounds and he just stayed with the sound of an A. So he would say the A sound in different pitches, but no other sound. And that was like a red flag. So immediately after that, pediatricians was like, okay, yeah, you need to check the baby. Something is not right. He's still not crying. He's not rolling. He's not doing tummy time. He's not trying to sit. It's like then, all the little things that made him a perfect baby made something that we need to worry about. This was very hard because we knew something, but as parents there was nothing that he can't like there was nothing that doctors would be like, oh, let's check for this because maybe it's this. And then he start therapies, still no diagnosis, so that's the way you go. You treat the symptoms, you treat what you have. So he started with physical therapy, occupational therapy. Um, he we we got him a special education teacher to teach him how to play, because he didn't interact with us. He didn't interact with toys. He didn't play with toys, and he was already 8 months old. And so and this like and he's keeping this therapy since. So we start therapy when he was 8 months until the day he just turned 3 years old. He still has a lot of therapies daily. Later on, we had speech, and later on, we had ABA, which is a behavioral therapy, and we just keep on adding things as we go. Then, when he was a year and a half, well, then of course, as he's doing all these therapies, at the same time, we are like, we need to see specialists. We went to see geneticists, neurologists, everybody, like, let's do an MRI, an EEG, an EKG, let's check everything. And we were able to, it's a longer story, but we're able to do the DNA sequence that led us to the diagnosis in December of 2022. And the paper, Medicine's paper, just came out like 2 months before. And yeah, and then on that night, we got the paper, and then it's like, okay, let's go search. Yeah. And then I think I'm just going over other but
No, that's
but
helpful to
this is Danny's
understand.
story. And he's still, he was a, yeah, he was a little walker. He was a everything. He's doing everything a little bit later, with the help of therapies. He's still nonverbal, and he doesn't have much receptive language. So cognition, it's where we see that he struggles the most. But we'll keep on working, and we'll keep on finding treatments, and getting to know the disease more to be able to help him further.
I think it's important to think downstream on the mechanism, because there are kind of three main phenotypes of this condition. Yeah, the developmental delays and cognitive impairment is one, and the epilepsy is another. And
Mhm.
like our son, fortunately has not showed yet any signs of seizure, which we're very thankful for. But there seem to be sort of these two, phenotypes, and they do not necessarily correlate. So sometimes, from our experience, sometimes you have kids with very severe epileptic phenotype, dozens of seizures every day, but they are more or less okay in the developmental aspect of things. And then, kids like our son, they don't have epilepsy but have a strong delay in neurodevelopment. So this kind of points to likely different downstream mechanisms, and this may be very independent, but we don't know.
Yeah, with further research, hopefully learning more about what else is leading to those two different phenotypes, because you're saying that most kids and adults, if there have been adults identified, fall into one of those buckets in terms of how the mutation that's been identified is presenting in that person. And okay, well, there must be something else that's leading to that, like you were saying with downstream effects. And I imagine when you guys were learning about this diagnosis, the first thing you do is go to Google. Okay, well, what resources and support are out there? And you must have come up somewhat empty, because that must have led to the start of the v-ATPase Alliance. So what are some of the mission and goals of the Alliance, and what was the driving force behind creating this? And I can't believe I haven't said this yet, but the fact that, Anna, you're wearing the zebra print shirt, I mean, that is totally to be noted in terms of rare diseases, and such a cool shirt also for you, Luis, of the care about rare. So, for people not watching the video, a very, very, perfect outfit for the day for both of you.
That's our Luis is wearing our official v-ATPase Alliance shirt. So, and but to the point, that I don't know if you want to go, Luis.
No, I think there were kind of two main things we went for Google.
Well, can I
One
just
was,
add one thing? Just one thing is that, so the good thing about Luis and me together is that Luis is a scientist and I Google, and then
yeah, but I guess this time both of us went to Google,
I
not to
know.
Google, and the Oracle, and but I think we were looking for different things,
Hmm.
and we couldn't find any of them. One was just try to find other families, because
Hmm.
this was new for our doctor. Like, picture this: we get a diagnosis in December that was first published, or sort of the association between the gene and a pediatric condition had been published like 2 months before, and so this was new even for doctors. They didn't know what to tell us beyond what was on the genetic report and reported on the paper. And so it was a very lonely start in this journey. We wanted to find other families. That was sort of one thing. See if this had been reported elsewhere, and the other one was like, I was I was perhaps a little bit in denial, and I was thinking that, okay, we got this diagnosis. It was published 2 months ago. Maybe there's something like the doctor is missing. Maybe there is some research on v-ATPase activators that they're not aware, because there was just not enough time, to process this, and for doctors to be aware of this condition. And so I was desperately looking in the literature for any v-ATPase activator, and unfortunately I came empty-handed, the same way Rita came empty-handed on trying to find organizations and patient advocate groups that could help us find other families. And so we started the alliance, and maybe Rita, go ahead and talk a little bit about it, and then I
Yes.
can pick up on research side.
Yeah, so we try to combine our both backgrounds, because I have a background in communication, and so the idea was to just go to social media and create all the content that we wanted to find out there. He's a researcher, so he can translate all those medical terms into lay communication, so we can literally change the fact that a person is Googling ATP6V0C or any other v-ATPase condition and come up empty handed. So on May 1st of last year, I started all the handles in all social media. I created content for about 2 weeks. Every day, every single day, a content would come out. Louise would vet everything that was scientific, so I would not make incorrect information available and not help anyone that way. And this is how we started. And the day before, I literally Google and print screened a bunch of databases and genetic databases and everything, and for Dennis gene, and everything came out 0 results. And so it's like, okay, tomorrow we're going to change that. And that's when we started. And it was an amazing journey, because this was just a social media project to, put information out there, because we had it, and we thought other people might be looking for it and not finding. And then in less than a week, I was talking with a family in Australia. And then in 2 weeks, 3 weeks, we were 10 families, 20 families, 40 families. A lot of people were Googling it and not finding results. And then in less than 6 months, we were about 50 families. So it just made sense that uh, this social media project was no longer just a social media project. We got involved with other families. We got to know them. We created a private Facebook group. We shared our story. Other families shared their story. And we also invited one of the moms to be like, hey, let's take this serious. Let's make a foundation, because uh, nobody else is doing anything. And just being in a Facebook group and having social media is not, yeah, it's great to find families. And yeah, please, families, keep reaching out to us and growing our community and sharing so we can learn more, but it's not enough. And so we decided that on November 1st, we register the v-ATPase social media project as an official nonprofit status official, and we're doing all the nonprofit things now.
And our goal is really to develop treatment. So
Yes.
it's a research focused organization. I want to point out that like Kara was saying, the v-ATPase is a complex, and so it has multiple subunits, and therefore there are multiple genes that contribute to that complex. And we learned in the process that the V0C is one gene that causes the developmental and epileptic encephalopathy, but it's not the only one. So there's a few other genes that have an identical clinical presentation, and so that's why we call it v-ATPase Alliance, because we ended up looking for families with these other genes as well, but a shared condition. And so we're kind of an alliance of v-ATPase genes, looking for for treatments, and for treatments, at this point we're still looking at all possibilities, because we want to have multiple shots on goal and see what might be useful in the shorter term and in the longer time frame. And so our research agenda, we plan to look at drug repurposing efforts, small molecule therapeutics. Interestingly, although there's virtually no literature on v-ATPase activators, there is quite a lot of research on v-ATPase inhibitors, because it seems that it holds some potential as a therapeutic for certain cancers. Now, when you go on a drug screening campaign for small molecule inhibitors, I mean, it's not uncommon that you end up finding some activators as well. And so this actually, I want to take this opportunity, to ask our listeners that if you work at or know of a company or a lab that has done some work modulating v-ATPase activity, please reach out, because that really might be our best chance. Who knows? And so we're really looking into that. And obviously, this is a very well defined genetic condition, and so obviously we want to consider strategies for personalized interventions with gene therapy approaches. There's still some groundwork we need to do. Like Kara was saying, I think a lot of research points to a loss of function mechanism, at least to some of the variants. But we need to dig a little more on that, because obviously that sort of directs us into the right therapeutic strategy. So we're really trying to consider all the different angles.
Part of the reason why we develop these assays in yeast, and I think a lot of people might have questions of, right, why would you study epilepsy or neurological disorders in the single cell eukaryotic organism that doesn't have a brain, right? It makes no sense. We got a lot of that back on our grain applications, but one of the useful tools goes back to this drug screening, right? you could screen drugs in yeast and see if you can, the phenotype we primarily looked at, but there's a couple different, was just general growth. And so we did that in assays where we grew the yeast over a couple days and measured essentially what we would call optical density. So how many yeast cells are in this little well of a plate we're using. But, there's opportunity to use the yeast constructs we have, right? They're on plasmids, so you can put them into multiple different types of yeast too, with patient variants, and screen a lot of drugs quickly. In the paper, a group that we collaborated with in Montreal actually created C. elegans or worm lines, and those are also useful tools that you can drug screen. So part of the work of the functional assays of saying how did these affect the v-ATPase in general was also, as a research group, trying to say how could we also develop this in a way where we could screen for these small molecules that might be helpful in the short term. They might not be the best, long-term answer, but they could be something that works. And so I'll put that plug out there that, if people are listening to, Luis can help get in contact with me if you can't find me directly, but we save all of those lines and everything, so they are available for research to send out if it's really needed.
Yeah, that is wonderful, and I think it's just so powerful to be able to have this alliance so that you are connected with so many families and people with one of these pathogenic variants, mutations, so that when the time comes for, say, natural history study, or there is a potential treatment that we want to be testing, you already have this group and growing database, really, of people that are ready to go in that sense. So it's such a needed part of any, I think, rare disease, especially when we talk about, I don't know if this would be considered ultra rare, but it kind of sounds like it's in that, category, that it's really important to have this. And it's just so great of seeing how nonprofits can connect with other researchers already working on things and collaborate and just see how more quickly treatments and everything can come together. So this was just really interesting. Was there anything else any of you wanted to leave our audience with?
That's a big question. Well, I got to say, to anyone affected with the v-ATPase, if you're listening because you got diagnosis, if you don't know us, if you want to reach out to us, you have all our information in the show notes. So you can just, pop an email, come to our website, come to our social media. We will have you. We will listen to you. We will learn from you. We will share what we learn. We don't know everything, but we are creating the structure to be able to support everyone already with a diagnosis and anyone to come. They will for sure have more information than everyone else who was diagnosed before us. That's a given, because there was no information out there, and now you can Google. Dr. Google has some information for you too. And so if you know someone with diagnosis, just reach out, and we'll be here.
Wonderful. Well, thank you again so much, all of you, for coming on the show. Really appreciate you opening up about your experiences and just really encouraging of all the work you guys are doing so that you can, make a difference. And, my well wishes to Danny and everything, as he, grows up and everything, of just having these connections with other kids and stuff, I think is going to be very valuable, not just for you guys, but Danny too. So thank you again, guys, so much.
Thank you.
Thanks.
Thank you.
Thanks for watching DNA Today. To access all of our episodes, head over to dnatoday.com. We also have a lot of bonus content on there that you can enjoy. If you have any questions, comments, suggestions, guest pitches, you name it, send them in to info@dnatoday.com. We'd also really appreciate if you could take a moment to rate and review the podcast on Apple, Spotify, wherever you listen to the show. It really helps more nerds like yourself find the show. Also, if you like giveaways and other ways to connect with us, I recommend following us on social. We're at DNA Today Podcast. We also have a Patreon if you want to be the most level involved in the show. That's also at DNA Today Podcast. Thank you so much for listening and watching. Join us next time to learn and discover new advances in the world of genetics.
We're all made of DNA. We're all made of the same chemical DNA.